Hello class, today we're going to be doing notes on cell reproduction in chapter 8. So we're going to be looking at mitosis and meiosis. If you happen to be looking at your notes packet, we're going to be going through chromosomes all the way up to the big dark line where it says types of cell reproduction. So we're going to go through the types of chromosomes, kind of how they're put together, give descriptions of those, and what they are basically. Why do we have them? How many do we have? That kind of thing. So again, this is section 8.1, chapter 8, section 1, and we're going to be looking at a couple things like this here. We have this as a karyotype, and you'll be doing one of these in class, cutting some of these out and looking at them just like a scientist would if they were looking at a particular individual to see if they had any disorders. And then basically what we're doing, this is the nucleus, and so we have chromosomes and chromatin inside of this nucleus. So we're going to talk about what those are and when they become chromosomes versus chromatin. All right, so on your notes packet, you can see you have a picture of this. If you're not using the notes packet, you may want to draw this out and put it in your notes as it is. So if we have two chromosomes that are identical in appearance, they are called homologous chromosomes. So notice these two are identical in appearance, the length, they have banding patterns, and what scientists will do is they'll band them so that they can see kind of where these chromosomes are, and they'll be able to identify them as homologous chromosomes. And the reason we have homologous chromosomes is because you got one of that particular type of chromosome from your mom and one of that particular type of chromosome from your dad. They are held together by this thing in the center called a centromere, and not the two homologous chromosomes, so take that back here a little bit, but the sister chromatids. So our two sides of the chromosome here are held together by a centromere. It's kind of like a little button. You could call it a little button. And then the sister chromatids are identified as the two legs. You can see that picture again there at the bottom, just to remind you and also make sure that you have it written in correctly. So chromosomes, a definition, they are a rod-shaped structure made up of DNA and proteins. And generally we think of just DNA, but there are proteins that help kind of wrap all that DNA up into a tiny little ball. Sister chromatids are the identical halves of a chromosome. A centromere is a protein that holds these sister chromatids together, and I like to often refer to it as like a little button. And then homologous chromosomes are chromosomes that code for the same trait that come from your mom and your dad. So you have a chromosome number 1 from your mom and a chromosome number 1 from your dad. The same thing's true for chromosome number 2 and chromosome number 3, and so on as you go through the list of chromosomes. Continuing on with some definitions, if you're using the notes packet, this will be down one more box. The chromatin is less tightly coiled strands of DNA. So when we have chromatin, it is not the uncoiled chromosomes that we think of. So chromatin is actually what your DNA or chromosomes look like when it is not dividing. When it's going to go through mitosis, we will see them become chromosomes. They kind of uncoil a little bit more and then become these chromosomes. So as chromatin, they're so tightly coiled you can't even see kind of the difference between one chromosome and another. They're really tightly coiled inside the nucleus just to kind of fit inside that nucleus. Chromosomes will occur during cell division. So the chromatin will uncoil and they make chromosomes. All right, for every organism, they have a specific number of chromosomes in their body. So we have 46, you can see that there, and you can put a couple other examples in if you'd like for your notes, but you're not going to have to, memorize those or anything, but if you just want to have an idea of what some of the other ones are, you can put those in. But each species has a specific number of chromosomes. So normal for humans is 46 chromosomes. Abnormal is anything above or below 46. There are individuals that can survive without having 46 chromosomes. They might have 45 or they might have 47. And there are other disorders that cannot survive. So if you're missing or if you have an extra chromosome number 21, that's Down syndrome. But if you're missing a chromosome, it could be ones that are fatal or it could be ones that would allow you to survive. So it just depends on which one it is. And we'll get into some of those in another chapter. You can see here that some things, like the fruit fly, only have 8 chromosomes, and that's one that's often studied in genetics because it only has 8 chromosomes, so there's not as much to have to look at. But then there's some things like an amoeba that has 50 chromosomes. So it doesn't necessarily relate to how complex that organism is. It's just how that organism is made and how many chromosomes it needs to have all of the DNA it needs. All right, sex chromosomes. So we're going to go through 2 types of chromosomes. Sex chromosomes are the chromosomes that determine whether you are male or female. There are other genes that are located on the X and the Y. So for humans, we have the X and the Y chromosome. There's things that are attached to that X and Y chromosome. But depending on if you have XX or XY will determine if you are male or female for our species. So XY means male, XX means female. And we'll get into some of that more in chapter 12 when we get to disorders and also some things that are specific, genetic traits that are specific to humans. Autosomal chromosomes are all of the body cells and all the body chromosomes. So these are all the other ones. These are the other 44 chromosomes, you'd say. And these determine lots of other things like hair color, eye color, skin color, maybe things that we've talked about in class like hitchhiker's thumb and widow's peak, and if you can roll your tongue and things like that that are going to be on all these other chromosomes. So this is autosomal chromosomes are anything that are not the sex chromosomes. So we have 46 total chromosomes. 2 of them are sex chromosomes, either XX or XY, and then 44 of them are the other body chromosomes. Okay, next on your sheet is karyotype, and you're going to be doing this in class as a little lab. Basically, a karyotype is just a map of the organism's chromosomes. And you can see here what scientists will do. They're going to take all of the chromosomes. They basically do this when the cell is going to go into mitosis, and so we'll talk about that when we get in that section. But when the cell is going through mitosis, they can see all the individual chromosomes, and so they basically freeze that cell at that particular time. They put in these kind of radioactive tracers that band the chromosomes. You can see that they have different banding patterns. And then they have this kind of jumble of chromosomes. So what they do, what you guys are going to do in class, you cut out each set of chromosomes, and then you try and line them up and pair them up correctly. And you do this by the banding pattern and also the length and the appearance of them, so that you can see if the individual has the correct number of chromosomes, and also you can see male and female. You can notice if there's any banding pattern that might be missing, so maybe there's a leg of a chromosome that's missing, or there's some other problem with the particular karyotype. And if there's a problem, then they can go and diagnose that particular issue. So a karyotype is very useful in looking at genetic disorders and trying to find out if there's a chromosomal abnormality with that particular individual. Okay, and the last thing that we're going to cover today in this set of notes is diploid and haploid. So di, if you look at the beginning of that word, di, that means 2. So you're going to look at 2 cells, basically. And haploid is half. And in this one, we've talked about cells. We're not looking at cells. I should have said that different. Di is 2, and then what we're going to be looking at is 2 sets of chromosomes, or haploid is 1 set of chromosomes. And the way to think of haploid, it kind of sounds like half. And so if that will help you, that might be a good way to remember it. So diploid are cells that have two sets of chromosomes, and they're represented by 2N. They're called diploid. So they have two sets of every single chromosome, and that's like the karyotype we just looked at. That would be 46 chromosomes. One set from your mom, one set from your dad. The haploid number of cells, or the haploid number of chromosomes, is 23 for humans, half of 46. And this haploid number is going to occur in egg and sperm cells only. The reason for that is that when an egg cell meets a sperm cell, they're going to make a full human that has 46 chromosomes. So we need half of them in the egg and half in the sperm so that when they come together and unite, they make a zygote, which is going to have 46 chromosomes. So the haploid number is represented by 1N and the diploid number is represented by 2N. Okay, that's where we're going to end today with notes, and that will complete chapter 8, section 1.